Welcome to BioMentors. Today our topic of discussion is from plant tissue culture series about somatic embryogenesis. So let's get started. In the last lecture we discussed in callus culture that in the end, the last step of callus culture was multiplication and morphogenesis, in which we can make either organs, which means root, stems and leaves from that callus culture, or we can go for the formation of somatic embryoids. So what is this somatic embryoids or somatic embryogenesis means? We will see in this video, so stay tuned and watch this video thoroughly. Introduction. So somatic embryogenesis is a process where embryo is derived from single somatic cells or group of somatic cells. Somatic cells, which means they are non-embryonic cells. Any part of the plant which is non-embryonic, for example, the cells of a leaf, is called as somatic somatic cells, not somatic embryos. And from that part of plant, we are going to make a embryo, and thus it is called as somatic embryos. And we will refer this somatic embryos by SE, capital S and capital E, and are formed from the plant cells, are not normally involved in somatic embryo formation, which we discussed. Embryo formed by somatic embryogenesis is called as embryoids. So the embryos formed from somatic embryogenesis is called as embryoids, and this process was discovered first time in Dicos carota carrot plant. This was first seen in carrot plant. Now selection of EXPLANT. The plant source material to induce somatic embryogenesis is very diverse. Many types of plants are used, but generally responsive plants which are actively dividing, such as carrots, are used for somatic embryogenesis. Radish, or any type of plant which can be easily propagated on the nutrient media are used. There are some plants such as cereals and legumes in which EXPLANT varies within the genetically identical species, and these plants can also be converted into somatic embryoids. Various types of EXPLANT used for somatic cells are immature zygotic embryos. They can be immature zygotic embryos or any other part of the plant, such as inflorescence. It could be derived from callus culture as well as cell suspension culture, from the plant petiole, protoplast of leaves, or protoplast, stem and roots. So it can be formed from any part of the plant. Now, steps of somatic embryogenesis. The first step is induction of somatic embryoids onto the nutrient medium. Then after the development of somatic embryoids, and the last is maturation of somatic embryoids, and then last is hardening of somatic embryoids. So this is the diagram of somatic embryoids. First we induce any part of the, we will take the EXPLANT and then we will put that on the nutrient medium, and here a callus is taken for the somatic embryogenesis. Then development of the somatic embryoids, and finally maturation of the somatic embryo is seen in the last two figures. These are the basic steps for somatic embryogenesis. Now steps, we will look these steps into detail. So first step is induction. Auxin is generally necessary to induce somatic embryogenesis, and generally the auxin which is used for somatic embryogenesis is 2,4-D. Relative concentration of auxin depends on the nature and the type of species which we are using. Development. Cell division and proliferation in presence of auxin is seen. So auxin is the main hormone to induce somatic embryoids. Then this cell is released into oxygen-free medium. Then these cells are transferred into oxygen-free medium. This cells is then these cells are in a cluster. First, these cells are in the clusters, and this cluster is called as polyembryonic cells. Proembryonic cells. This mass or this cluster of cells is called as proembryonic cells during development stage. Then maturation of the callus culture. So after putting this culture into the new nutrient media, they go through the final stage, which is called as embryogenesis. And during maturation of the embryos, because this embryo do not contain a reserve food material, endosperm, they do not have endosperm. Normal seeds have reserve food material in form of endosperm. So nutrition, they lack nutrition for the developing of embryos, and thus they remain small in size and the growth also declines. To avoid this, somatic embryo remains. To avoid this underdevelopment, ABA is used, which is abscisic acid is used. So why this abscisic acid is used? Because abscisic acid will prevent precious germination and promote normal development of embryoids by triggering gene expression. Normally, ABA induces somatic embryogenesis during drying down stage of the seeds. And here the same process is repeated by adding the ABA onto the nutrition medium. But still, if we add ABA to this solution, there would be a low germ inability or conversion into plant is very poor. So outcome of this somatic embryoids are generally very poor, and this is the main drawback of somatic embryogenesis. Types of somatic embryogenesis. So there are two types of somatic embryogenesis. The first is direct somatic embryo formation, in which shoot is formed first, then after shoot elongation is seen, and finally the rooting of the embryo is seen. When the embryos are formed directly from the explant, so from the explant directly the shoot and the roots will form, and they are identical without formation of callus tissue. Such type of somatic embryogenesis is called as direct somatic embryogenesis. We don't need any formation of callus. This explant capable of direct embryogenesis, those plant which has this capability are called as pre-embryonic determined cells. And they have this potential to form somatic embryo. These cells are committed to embryo development and need only to be released. So these plant cells are very unique, and they just need a nutrient media, and then they are released to the fields. Second is indirect somatic embryogenesis, where callus is formed from the explant, developed, and this embryo are called as IEDCs. Indirect somatic embryogenic cells. So in this type of somatic embryogenesis, indirect type of somatic embryogenesis, callus formation is seen, and then after shoot and root formation is seen, and then they are released onto the field or to the greenhouse. All right, callus is formed in this type of somatic embryogenesis. So what are the disadvantages and advantages of somatic embryogenesis? The main advantage of the somatic embryogenesis, that lack of material is not there is no lack of limiting material for the tissue culture. High propagation rate. For some species, it has high propagation rate. Somaclonal variations, genetic variations can be done with the help of somatic, somatic embryogenesis. Good, it is very good for germplasm conservation, for the conservation of germplasm. Labor saving, it is labor saving and eliminate disease and viruses in the plant. It can eliminate disease and viruses in the plant. We can also conserve rare and endangered species by this method. And lastly, we can also germinate such type of seeds which do not germinate normally. Embryogenic seeds do not germinate, such as the seeds of orchid, right? Disadvantages: confined to very few species. This confined to very few species. The somatic embryo shows very poor germination rate because they lack reserve food material. They do not have reserve food material, and thus the germination rate is very low. And second is physiological and biological immaturity is there in those plants. The somatic embryos or somatic embryoids could not be cultured for a long time period. So they should be shifted. And this is the main drawback for or the limitation in commercial exploitation and mass propagation of somatic embryoids cannot be formed. So this was the main disadvantages. And this was the end of our topic. In the next video, we will discuss about artificial seeds.
Thanks for watching.